I'm here to talk about JMP Clinical 17. I'm the Senior Product Manager at JMP for Health and Life Sciences, and JMP Clinical is one of the products that I'm responsible for. For those of you that have not seen the product before or curious about it, I'll tell a little bit about what JMP Clinical is. It's a focused and specialized product for clinical trial data review. And we design it to give users straight out of the box functionality to do thorough reviews of clinical trials at the study, site, and subject level. And we've utilized, and this product is utilized across the pharmaceutical industry and in several regulatory agencies. So we're pretty excited about showing you this. So one thing that enables us to write this and develop this as a product is that we utilize a data standard. We follow the CDISC data model, and we design our product to be used with data that conforms to that CDISC data model. So if you have data that's in the SDTM and ATOM format, you can load that directly into JMP and start going right away and doing reviews. These data standards are mandated for new drug product submissions in the US and Japan, and they're recommended in many other regions as well. And having a well-defined data standard, data model allows us to develop the standardized approaches for analyzing it. So that's the real value to the industry and what enables us to be able to deliver the product the way we do. And so we've been at JMP involved with the CDISC organization for many years. And actually right now, Dr. Wenjun Bao, who Gail mentioned earlier in the intro, she is one of our senior developers, and she's on the board for CDISC. And she's actually over at a board meeting this week for CDISC in Europe. So we have a great relationship with them, and they appreciate what we've done to help further the usefulness and the utilization of the CDISC standards in the industry. So JMP Clinical 17. It's all new. It's a fresh approach. So it's been re-engineered to be a JMP-only product. The previous versions of JMP Clinical utilized SAS for data processing and analysis. So when you got JMP Clinical, it actually came with a JMP and the JMP Clinical package plus desktop SAS. And it was a large installation package. It was difficult to manage and deploy. And it was just a kind of a big, unwieldy product for us to manage. And so it was time for us to update the product to utilize JMP only. And it's been a major effort to do that. It's almost been a complete rewrite of the JMP Clinical part of the software. So it's a leaner, simpler, and faster product now. It's a smaller installation package, 12 times smaller in the actual install package size. So that makes it easier for deployment and fast installation. It's simplified in many ways. You'll see that if you've used Clinical before, the reports just run now when you launch the reports. There's no intermediate dialogue that you have to specify a lot of settings. The reports just run with default settings, but there is an option to change those settings if you want to. And then we have a number of ready-to-use clinical review reports. And another thing that's nice is these reports can also be published to our product JMP Live. So if you get access to JMP Live 17, you can publish to a web-based viewer the reports that you generate in JMP Clinical. JMP Clinical is available for Windows. It has always been a Windows product, but now it's also available for Mac OS because we no longer need to use SAS, and SAS did not run on Macintosh OS. And another new thing is JMP Clinical 17 also now comes with JMP Pro 17. So Clinical's architecture is that it's JMP plus the clinical package that comes with, that we develop in JMP scripting language. And in the past, we provided it with JMP, but now we're providing with JMP Pro, which has more advanced analytics features that you can utilize on your own to go further with your clinical data beyond what we've provided in the standardized reviews that we put into the clinical product. We're delivering this in a staged approach because it's been a very aggressive timeline for us to move to this new version, not using SAS. We delivered in November version 17.0, and it had 30 reports in it that were ready to go, mostly for safety reviews and medical monitoring. And just shortly, within a week or two, we're going to release JMP Clinical 17.1, which adds more reports, including a new FDA medical query and algorithmic FDA medical query reports, which I'll show you. And then we have some additional reports that we haven't had time to get converted over yet that we will release in September when we release JMP Clinical 17.2. And along the way, we're making improvements in performance, and if there are some bugs that need to be fixed, we're fixing those as well. So so that's all, that will be there. So by the end of the year, the complete new version will be there. We've got in 17 in this new product, we've got the FDA medical query and algorithmic medical query. We have study flow diagrams, which is a new report. We have updated risk reports for events and interventions. And the patient profiles report is now a standalone report. And so I'm going to show all of that to you, but with a demonstration here. So I'm going to exit out of there. I'm going to now share my desktop, just the whole screen. Okay, and I'm going to move over into my virtual machine where I have JMP Clinical running. I just do this it's a little bit easier for me to manage all the versions of JMP that I have to look at as a product manager. So I have JMP Clinical installed on a virtual machine here. So so when you launch JMP Clinical, it looks very much the same as it did before. It prevents the JMP Clinical window with three major areas you can do. You have the area for studies, reviews, and settings. And where you start with JMP Clinical is you have to load data. We ship JMP Clinical with an example data called Nacartapine that's publicly available that we use for many years for demonstration purposes and for testing. And I'm going to use that study in the things I show you today. But I also have another study loaded. This was from a CDISC pilot that was done. And the way you load a study is you simply go to the Add Study button, and you have to give the study a name, and then you have to specify the folders where the SDTM and ATOM data are. And the minimum is you have to have at least one of those. You don't have to have both, but you at least have one, and you can have both if you have both ATOM and SDTM. I'll just open up one of those directories. You can look at it direct here. And all we have is those files there loaded. In this case, I have them as JMP data tables. The input format you can use are SAS data sets, either export, SAS export, or SAS 7 BDAT files, or you can use JMP data tables as the starting data. In this case, we're using JMP data tables. And then once you have the study loaded, there's a number of things you can do. One thing that's newer is we've added now the ability at the study level to set preferences for the study. So, and those preferences include the default settings for each of the analysis reports, or for many of the analysis reports. If you're familiar with the previous version of JMP Clinical, which was JMP Clinical 8, by the way, I forgot to mention that. So we went from JMP version 8 to JMP version 17 for JMP Clinical. The reason is we're syncing up our numbering convention for JMP Clinical to be on the same number as the rest of the JMP products. So JMP 17 is available, so this is JMP Clinical 17. But all of these settings used to show up in the dialog in JMP Clinical 8 that you had to specify and then run the report. Now you can set those as a preference. And so if you have certain preferences you want, you can set those here for many of the study reports. The other thing that we've had for a long time is you can also define the order and coloring of different categorical variables that you're going to use in the study. So for instance, for the treatment arm, you can set the preferred color you want to use for the treatment and placebo. And if there's multiple treatment arms, you can specify all of those. And so you can set those the way you would like them to be. And once you've got that all set up, then you can go and you can start doing reviews. I'm going to start out just doing a review manually. And I'm going to start with some of our study level reviews, just looking at overall the study. And I'm going to start with looking at a study flow diagram. This is a new report. This is also sometimes called a consort diagram, named after, I think, the consort organization came up with this. But this is just a high level summary of the flow of patients through the trial. So you can see how many were screened. In this case, 906 patients were screened. How many were actually randomized or excluded? 4 patients were excluded for whatever the exclusion criteria or inclusion criteria was for this trial. And then of the 902 that were accepted in the trial, they were randomized and into in this case, all of them were randomized into the treatment groups, and roughly half and half were distributed or randomized to the treatment arm for the actual treatment and the placebo arm. And then you can see a summary of the disposition of those patients in those arms, and in particular, how many completed the study, how many died, and how many were lost to follow up. So that's a nice high-level summary, and this type of diagram is generally expected, and oftentimes in a regulatory submission that you need to put that in just to provide that summary. Then we will look at maybe the demographics in the study, because one thing you want to check to see is what do the demographics look like in the study. What was the proportion of males and females, of the distribution of racial groups and ethnic groups, where the study was done. In this case, it was only done in the United States, but if it was a multi-country study, where that happened. Look at the distribution of the ages of the study participants. And so you can look at all those. You can actually then also do a comparison between the two treatment arms. So, and that's a way to check to see, was the randomization reasonably good? So in this case, we're looking at, is there a difference between the mean ages? And we can actually from here even turn on a comparison of the distributions of the two distributions of age across the treatment arms to see, are they comparable or not? And that's just a way to check how good was the randomization, or was there some issue, but maybe potential bias is put into the study because the demographics weren't balanced evenly across the treatment arms. And so in this case, we've got that you can look at it both for the continuous and categorical variables. So this is a contingency analysis and a mosaic plot of the distribution of sex, of gender across the two treatment arms, and they look roughly the same, pretty close. So then you can also then move into some other types of reports that are more study level. So you might want to look at not just the you might want to look at the study visits, and this is just an indication of the trial progression, how many people were moving through the trial by visit number. And it gives you an idea of different things, like were the two treatment arms, roughly staying proportional across the number of study visits. And if they're not, that could be an indication that there's something going on with either the study arms, patients dropping out more frequently or not. You can actually do another way to look at this is I'll run this again, do the same study visits, and let that generate. And now what I'm going to do is I'm going to turn on the option to add a by variable. And in this case, what I'll do is I'll do this by, let's do this by sex. So I'm going to scroll down here and find sex. There we go. Click OK. And now what it does is it takes that same graph and it splits it out by that demographic variable. And now I can get a comparison of the progression of patients through the trial over time by the demographic variable. And again, you're looking to see, does the shape of those profiles across time look similar for the two demographics or not? If not, that could be an indication that this demographic variable maybe has an impact on the study. And it's also just a measure of the study health. So you've got lots of ways to do this. You can one thing's a little not so nice about this the way it is right now is it doesn't really describe the tab here doesn't really describe the analysis so well, but we've got the ability to go in here and manage the tabs, and you can change the name. So I'll just change the name of this to be study visits by sex, right? And click OK, right? And now that's more descriptive of what that tab is. So this is a nice level summary of what's happened in the study at the high level. And you might want to share this with people. And we've got the ability to publish this out to our JMP Live server. And I want to show how you can do that. So I'm just going to create this and publish this out. This is on our public site. So I'll keep this out there. You'll be actually able to go out and see this after it's published. But let me just give it a moment to, there we go. So now it's publishing. and give it a second to load up in the browser. And it will show a HTML version of that output that was created. There we go. Yeah. So again, it's got all of those reports there, and you can go through them one at a time. There's the study flow diagram. There's the demographics distribution. There's the study visit report. And there's the study visit by sex report. Okay. That's a nice way when you create something within JMP Clinical, you can share that with more broadly with others in your organization if you have JMP Live. So let me go and show another type of review, and I'm going to let this run. This one takes a little bit of time to generate because it's doing different types of things. I'm going to let it run, but I'm going to talk while it's preparing about some of the new features we've added, which are FDA medical queries and algorithmic medical queries while that runs. So these are actually slides that the FDA put out recently. You can just find these online, and I'll put a link to it in the presentation. But these talk about what FDA medical queries are and why they're important. So these are FDA medical queries are more they're not more complex, but they are medical queries that are different than the standard medical queries that based on MedDRA preferred terms. And they group together multiple terms often. So for instance, anxiety, right? In the MedDRA preferred term list, there are a lot of terms that might be related to anxiety, like panic disorder, panic attack, nervousness, generalized anxiety disorder, anxiety. And one way to say, well, was there anxiety in general, is to group all of those together. And that's one of the ideas around an FDA medical query, is grouping things together that are more granular into more general categories. Okay. And the FDA published a guidance for this recently, a draft guidance, and so we have that in JMP already because it was pretty easy for us to implement that in JMP once that came out. So we've got the FDA medical queries, and we both got the narrow and broad versions of them. So narrow are more specific to the medical concept, and broad terms kind of cast a wider net. So they're more like, well, maybe there was this, it's more like a 30% probability that the medical concept that it's included in the FDA medical query grouping occurred, whereas the narrow one's more specific, more like a 90% chance that it included. The other thing that we've added are algorithmic FDA medical queries, and those are medical queries that are similar to FMQs, but they also include other things from other domains other than just the adverse events, things like laboratory results or concomitant medications. And so I'll show you an example of that would be for hypoglycemia. This is an algorithmic medical query for hypoglycemia. So if a patient meets any one of these, they would be classified as meeting this hypoglycemia algorithmic FMQ. They have any of the hypoglycemia FMQ narrow terms. They have a plasma glucose less than 54 milligrams per deciliter, or they have any hypoglycemia broad term or supplemental term plus a plasma glucose less than 70 within the start date of 1 week, and or greater than 2 occurrence of hypoglycemia broad terms or supplemental terms plus greater than 2 occurrences of plasma glucose greater than. So you say, well, that's really complicated. Yeah, it is. It's a more complicated algorithmic approach for identifying potential adverse events, and it taking into a broader range of observations across multiple domains. And this is what the FDA has put out there. And so if you download the FDA medical query file and you put that in the right place for JMP Clinical to use, you can actually create those. And so I'll just go ahead and show that right away. So here's the FDA medical query risk report, and I'm going to focus this down a little bit to where the rates of incidence were higher for these. So I'm going to look at where the rates of incidence were at least 2%, which is a much smaller group of terms. And you can see that in this study, there was a lot of evidence for hypotension, heart failure, systemic hypertension, or one of the ones that are the most common. And the way this risk report reads is we have on the left-hand side of the risk report plot, we have the actual percentages or frequencies of the occurrence of that event in the treatment arm. So this is for placebo for falls. For falls, in the placebo group, it happened 18% of the time, and in the treatment group, it happened 35% of the time, right? And then we calculate the difference in those percentages with a confidence interval and create this forest plot that you can see, are those confidence intervals different from 0 altogether across all the terms. We also have a table of all those results below that's formatted nicely for you to be able to put into your reports. Okay. And this formatting is new for the tables, by the way, using some new features that are in JMP, and it's in the Tabulate platform that JMP has. Okay. So that's kind of the FMQ. We also have the algorithmic FMQs. And in this case, there were only a small number that actually of the FMQs where they were actually showed up as being possible in this study, and none of them were statistically significant, but at least in terms of absolute difference, hyperglycemia was one of the ones that's potentially there's an elevated risk. And if the risk difference is positive, that means it's higher in the treatment group. If the risk difference is negative, it means it's higher in the placebo group. So in this case, these are all higher in the treatment group, the cardipine treatment arm. So that's kind of, we're really excited about this, that it's available in the product. I hope that by itself can be of interest to many people. Some other things you can do for adverse events include, you can look at just the overall distribution of adverse events. And I've created this report with some settings. Now this is a little bit different if you've used the previous version of Clinical. It used to be if you wanted to do this, you had to specify everything you wanted in a dialog and click run, and it would create the report. Now you can change a lot of these things just by changing things dynamically in the report. So for instance, I chose to stack the bar charts, have a stack version of bar charts where it shows the serious events and non-serious events, but I can change that and say, I want this by the intensity of the severity. And now it'll do it by mild, moderate, and severe. I can also just say, I only want to look at severe events. So I can just filter right away, and it only shows me the distribution for severe adverse events, and it just updates automatically. So that's a lot faster, a lot more dynamic and interactive. We have our incidence report, which this is the incidence or the counts of the adverse events, but scaled by essentially time on treatment, right? So it's more of a rate of events per number of days on treatment. And that's just a different way to view the rates, a more balanced way, particularly if for some reason in one treatment arm, patients were on treatment longer than in the placebo arm. Perhaps they were getting cured quickly and they were coming off, they were getting better faster. You'd want to adjust for that type of treatment, that type of difference in time scales. We have our new adverse event risk report. And again, I'm going to filter this down just to the more in absolute terms, the more commonly observed adverse events. And in this case, I have this grouped by the dictionary derived term. But I could change that and say, what I really want is I want this grouped by a high level group term. And it just reruns and it creates that. And again, I'll filter that down to the more common ones. And you can see that. And in this case, you can see in the treatment group where the risk differences are positive, you see things like procedural related issues, non-specific blood disorders, urinary tract signs and symptoms, heart failures are more common in the treatment group, but in the placebo group, you see vascular hypertensive disorders as the most common one, along with arteriosclerosis, stenosis, and vascular insufficiency. Now, in this case, this study was looking, they were testing the effectiveness of nicardipine for subarachnoid hemorrhage, so brain bleeding, brain swelling, and you expect patients that are in the placebo arm are going to experience those things, that they're having that actual medical condition. Those types of hypertensive disorders would be more common. You can also look at things like the summary of treatment adverse events, just how many subjects had treatments of adverse events, were they severe or not, and have a nice summary table of that. So that's a nice thing to be able to check to see how many of the adverse events really seem to be due to the treatment or not. You can get a summary of the exposure. This is just how many days you were being treated in the different treatment arms. And again, this is just more of a study health thing, but also an indication, were patients basically being treated for the same period of time in each treatment arm or not. And you can do a comparison just looking at this bar chart by days. You can look at the means and standard deviations. You can do a look at the number of doses that patients received across the two treatment groups. And just like I did before, this is an additional JMP feature. You can turn on, the compare densities to look and just see what's the distribution of those number of treatments similar across the treatment arms in this case. You can go a little bit further if you want to look for some very specific things like potential liver injury. You can use the Hy's law plot, which is a heuristic rule that's designed to identify patients that are potentially having severe liver injury with elevated liver enzymes and bilirubin, things like that. And and this is really nice. You can just select those patients, and you can see, okay, those are the patients there, and you can actually zoom in on those patients later. I'll show you in a minute another view of this where I've zoomed in just on the patients that were in the active treatment arm that had an adverse event or that had evidence of Hy's law, and we can look at them a little bit more closer. The final thing is we have the ability to create narratives. So this is something that happens a lot. And what I'm actually going to do here is I'm actually going to, since I've selected just the Hy's law patients, the ones that have potential liver injury, I'm going to actually push that selection into the review subject filter. And this is something that's new, a little bit newer too, the way this works. So now what I've done is I've created this overall filter that applies to all of the subject level reports that I've created. Some of these reports are not subject level, so they're not updated, but the ones that are subject level get updated according to this study filter. And so now it's just showing those patients that are potential Hy's law. And I'm going to go over here and I'm going to create narratives for those. And I'm actually going to, in the narrative, I'm going to turn on also the ability. I want to look at the, I've got the exposure and I've got the concomitant medications turned on. So those are the things I wanted to see anyway. And I'm going to run that report. And so it's going to take all the data from across the domains that are related to these patients and create a narrative in this case, in a Word document format that I can look, and it's just a nice summary of the patient. I know that's something that has to be done for medical monitoring and medical review. And so we make this really easy to do. These templates are also customizable if you want to. So you could customize them to meet your own format and use your own template. Yeah, the ones we provide are reasonably good and a good place to get started on generating these reports. All right, so I'm going to stop there for a second, see if any other questions came through before I go any further.
No, Sam, you can continue. Thanks.
Okay. So let me go in a little bit further. I'm going to a different virtual machine because I set this up differently. One of the things is some of these things do take a little bit of time to run. And so demonstrating them live, we would be sitting and waiting 30 seconds for things to update. And it's not, it's for you working, it's fine. But for doing a demonstration, it's just a lot of empty time. So I decided to go ahead and run this Hy's law screening again, just selecting the Hy's law patients, but also using the review subject filter and only choosing then on top of that, the patients that were in the active treatment arm, the nicardipine arm. And I've created patient profiles for those. And those patient profiles are a high level summary of just everything that happened to the patient in the study. So this is for one patient that was on treatment. You can tell what visit numbers they went to, their date that they were randomized, the exposure they had. So they were dosed to nicardipine at several visits. You can look at their adverse events and the duration of their adverse events. You can look at the concomitant medications that they were on and what's in the data that tells about the duration that they were taking it. And also in this case, I added in their lab results. In this case, the lab results that are indicative of Hy's law are the alanine aminotransferase, the alkaline phosphatase, the aspartate aminotransferase, and the bilirubin results. And so that just shows that, yeah, whatever that at a later time point, like at the study visit number 8, they had when their liver enzymes were measured, a big jump in those, and that's an indication that they were having a potential liver injury. So all of that's available graphically. We also have their medical history shown here on the right-hand side, along with the summary tables that describes all of those things that were shown in the graph. Okay. And then you can switch, if you wanted to do another patient, you could switch that. Because this takes a little bit of time, we force you to click on an apply button, and it's going to rerun and regenerate. And this, you know, it may take 30 seconds. And this is a little bit faster. So boom, there's another patient. Okay. So that's really cool. It used to be patient profiles was available as a drill down in other reports in JMP Clinical. Now it's just a standalone report. So you can just do patient profiles and on their own without having to run any other report. All right. Let me close this review that I had here. And now I'm going to show you a couple of the others. So I'm going to look at some reports we've created that we have for findings. And these are things like laboratory results, vital signs, EKG, anything that's sort of a measurement or a lab result or measurement you take on a patient. And we have things like distributions, box plots, mean change from baseline tables and plots. And so the time trends is the mean change from baseline versus time, and then some shift plots that are looking for just the things, how are patients shifting during the study, and then shift tables. And I'll describe each one of those briefly here. I also have waterfall plots. And again, I ran Hy's law screening again, which is another way to look at findings. Okay. I ran this with a saved template that we have. So you can create these templates that you can use, and you can actually use them on any study once you've created the template. So in this case, what I've got is, this is the mean change from baseline for lab results by study visit and grouped by the 2 different treatment groups. And you can scroll through each of the lab results and look for evidence of, you know, trends that might be happening by study visit, also potential outliers. Like there's a result here that's really, in the placebo group, someone had a really, really huge alkaline mean change from baseline for alkaline phosphatase, which that could be a data quality issue, that could be a medical issue, something to investigate a little bit further. And you can scroll through each one of those interactively. Okay. You can also change the domain that you want to look at. So if you'd rather look at, say, vital signs, you can switch that, and then it'll just switch to the vital signs domain. And now you can look at things like heart rate and blood pressure. Okay. We also have the ability to just look at the general distribution of lab results. This is more an indication of maybe study quality as well as the progression of the study. But, you know, are you like, you know, are how many by visit number, how many of these results are actually there? And you see for this, in this case, for the lab test results, you know, visit 1, everybody's getting all of the tests. But as you go through visits, patients are dropping out, and so you're getting fewer tests by study visit. And you can look and see also which tests, how frequently they were, what percentage of them were in the normal, high, and low range across the treatment groups, what the percentage of lab results were, making sure that both arms are being evaluated the same. And then you can also look at reference range indicator counts. So where they things normal, low, and high by study visit for the different lab results or the findings results here. And then you can dive a little bit deeper and look at, you know, just the distribution and differences in the distribution of the lab results in standard units across the treatment arms. So really a lot of information in this lab test results distribution. And again, you can change the domain if you want to a different domain. You can also choose different normalization if you want to, different normalization methods, and you can also choose whether you want to transform the results maybe to like a log 10 or a log 2 transformation. We've got mean change from baseline table, which is just a summary of by visit for the different lab results. What was the average mean change? What was the mean change from baseline? And we've also got a plot that shows a similar thing. This is mean change from baseline by study visit. And again, you can do this by the different findings results. And in this case, it has both the mean, but it also has all subjects. So when I look at the blood urea, the BUN, the blood urea nitrogen, you can see there's one patient here, a couple patients that have, an elevated increasing, and I can select that. And, I could push that to review study filter and just focus in on that one patient that had that high result if I wanted to. We've got the shift plots, which is just a plot of the baseline versus the mean post baseline result. And again, this is if you see things that shift, then things will not fall along the horizontal reference line, which is the y equals x line is the black line there. And you can look at, things like, are things generally falling along that black line or not? And if they are not, then that's indication that things are shifting from baseline as you move along through the study. And again, you've got shift tables, which these are using the categories of are you low, medium, high on the different findings results compared to the reference range indicator. So your low, medium reference range, low, medium, or high, low, normal, or high, and just a count, a frequency count. And that was actually shown in a graph in the time trends plot there. We've got waterfall plots, which are common plots that are used in this area. The way you interpret these is you're looking for differences in the distribution of the colors across the waterfall. So if you're seeing, in this case, the treatment arm is dark blue and the placebo arm is gray. And if you're seeing the grays and the blues sort of evenly distributed across the waterfall plot, that means that in this case, blood pressure roughly across all the subjects is the distribution's about the same. But if you're seeing more grouping of one color in one direction and another color in another direction, then that's an indication that those distributions are not the same. And people who've used these a lot kind of know their eyeball is trained to go, oh yeah, that I can see things look okay or they don't look okay, and I need to investigate further. And then of course, Hy's law screening is there as well. So that's another good findings report. All right, so I'll stop there for a second, see if any questions came through, and then I'll wrap up with just a couple other reports, some data quality reports that we have.
Sam, it's Gail. I'm not sure if you're going to get this. I think people would be interested now that I've really understood my study. Are you going to talk at all about submitting results?
Yeah, so, there are different ways to capture the results that you generated. One is you can just use JMP. You can copy paste, right? You can select things from the report and you can paste them in the document. The narratives, the adverse event narratives is another way to capture, on an individual subject level and other grouping levels, the summary of the adverse events that happened. You can also just output the results. So we have the ability to create a static report for the reviews. So you can create that in a PDF or you can put that into a rich text format, and it'll output it in a format that would require in general some further formatting, but it gets you really close to the output that you would want to create, or the doc you want to create, or at least something you can copy and paste, put somewhere else.
Thanks. That's the only question I'm seeing.
Okay, good. Well, let me wrap up here, and I want to show you some of the other reports we have. So I think the ones that we've got reports that are based on events that happen in the events domain. You can create a DSER-PSER report, which is just a summary report of the safety events that happened in a study that you might use for your regular reporting. So, and you have the ability to set like different cutoff dates if you want to choose a cutoff date, and some other, and do you want to choose particular event types or not? And again, it just captures a lot of the information you could get from other reports, like the baseline demographic characteristics, the summary of dispositions, and summary of exposure, the route of administration across the treatment arms, and then a tabular summary of the adverse events, and then a tabular summary of serious adverse events. And again, you could create this and make this into a report, right? So I'm just going to create that a report, just make that an RTF, and it's just going to create it. and there's your report. And you have to go in and, these are tables, and you'd have to go in and format the table a little bit and resize things if you wanted to make it a little bit nicer and cleaner. But yeah, it's there, ready to go. The last thing I'll show you is some of our data quality reports, and we're going to add more data quality reports in version 17.2. Additionally, we're going to have some oncology reports that we had in version 8. Data quality reports are these are more for study monitoring and looking to see are there issues with the actual data itself. So you can look for evidence of constant findings, right? Constant findings is when, someone's just copying over the last result or not, or maybe just being careless with results or made a mistake. It could also be evidence of fraud. And so there are things you can look at duplicate records, and sometimes those are bookkeeping errors. Sometimes they are also evidence of patient or study or site fraud. And they're also, other indications of study quality. You can look for missing findings and the number of missing findings there were across the different findings groups. And then you can also look at digit preference. And digit preference is kind of interesting because there you're either looking at, is the data being recorded correctly, or are people just making up data and putting in false data? And that one takes a little bit extra time to run. So it'll finish here in a few 20 or 30 seconds. There we go. So that's done. And then we have outliers reports, and these are univariate outlier reports. So you can look at for different findings to see are there potential outliers. And then we also have multivariate outliers where you can look across subjects and see are there subjects that are potential outliers. So let's just go through these one at a time. Constant findings, we're just looking to see, for instance, what parameter had the same finding result over and over and over again. You can click on that and you can see that was across different study sites. That bilirubin was the most common one across different study sites, across the treatment arms. If you saw this and you clicked on bilirubin, you saw it was like just one site, you go, oh, whoa, that's an issue with the site. We've got to go look at and see, are they following the protocol or doing other things? Same thing with duplicate records, a very similar thing. Here it looks like study site 29 had the most evidence of duplicate records balanced across the two treatment arms. Again, might be something to investigate further. Missing findings, this is more of a statistical plot, but what you're really looking for, and I'm going to just focus on like a particular, in this case, laboratory result. And you can look at this and say, okay, for this particular one, so this was at study site 18. We want to go maybe check on that, and then we could dive into the data a little bit deeper and select that one. You can actually view the data tables here. And so if I select that result is actually selected in the table, and I can just go to the jump menu and choose to say, give me the next selected result. And you can say, okay, that you can see what result it was, what study it was. You can actually go look and see and the missing findings. You can find out, you can find a little bit deeper where that came from. Okay. Digit preference, again, is just an indication. I'll just focus in on like the bun result and select the one here that's the most, seems maybe the most outlying or most unusual result. And clicking on that shows a drill down plot that shows these was the distribution of the last digit that was used. Okay. And to see that is that a bit unusual or not. In this case, it might be. There's lots of 9s. I think that's a bit unusual. Lots of that, the last digit being a 9. If I click on this one, I know there was one. Oh, I know what it was. It was pH. Let me go find pH here. This is a good one. You can see this. So I click pH, and I'll select some of these and drill down. And that one's not so interesting, but this one or this one. One of these, what it showed is that the digit 0 and 5 were the most common that were showing up. And so, but it was an evidence of people were rounding, rounding the results too frequently on, I forget which result it was now. There's one of those, but you can look and see, if you're always saying like the last digit is 0, 5, that means they're just rounding it off and maybe not recording it to the precision. Or that may just be the way the lab test is designed to report to that level of precision. This outlier report, again, looking at by a particular findings results. you can find, for across different study sites, those findings are there results that are outliers. So there's one here that's a potential outlier that for subject 141048, we might want to look at that and see, is that a medical result? Is that a data error? And then multivariate outliers, this is a little more statistical, but what it is it lets you use different levels of this nearest neighbor algorithm to say, how close is this point to its other nearest neighbors? In this case, how close is it to its eight other nearest neighbors? And you can find a point, and in particular, here's one that's a little bit at study site 20, the subject, a little bit of an outlier. And if we do it, with a fewer nearest neighbors, it even appears to be a little bit more outlying. So again, just a way to identify potential outlying subjects in a study. Also, it'll show you here how many results were missing and just more detailed statistical analysis at the bottom, but the graph is usually what you want to look at. All right, so I didn't cover everything about JMP Clinical. There's more to it than what I showed, but I hope I gave you a good overview of some of the things that are new, the new interface. For those of you that haven't seen it, what the product is capable of.